Okay, lab 23. This one talks about the different types of radiation and ultraviolet rays. Yeah, this was the one where we put, you inoculated your plate and then you covered half of it and put it under the UV light, and then we did either dark repair or light repair. So sometimes, I mean, he'll ask you things in the background, so you definitely want to read through like the background of each lab. Do you guys know the difference between ionizing and non-ionizing radiation? Because it talks about that in the background. X-rays and gamma rays, are they forms of ionizing or non-ionizing radiation? Yes, Lisa.
Ionizing.
Yeah, that's right. It's called ionizing radiation because their principal effect is to ionize water into highly reactive free radicals, apparently. So what we used in the lab, though, was non-ionizing. And let's see. Okay, can you guys tell me which UV ray is the most lethal out of UVC, UVB, or UVA? Probably want to know.
I want to say A, but I'm not sure.
Actually, A is not.
Oh, dang.
A has the longest wavelengths, and in the book it says that the shorter wavelengths have more energy.
It's a good thing I'm here.
So UVC is actually the most lethal because it has the shortest wavelengths. And then I would definitely read through this because it talks about the difference between light repair and dark repair and how it actually it restores DNA. So do you guys know, I mean, other than just putting the bacteria in the light and the bacteria in the dark, do you know the difference between light repair and dark repair? It's a little bit complicated, but he might ask you about some of these terms. So light repair, okay, so what light repair does is it activates photolysases, which split thymine dimers. And thymine dimers are what cause DNA mutation. So that's how light repair works. And dark repair uses endonuclease to repair the DNA. So I would definitely know photolyase and endonuclease. And these terms are actually on, I did an extra one for this lab. I just did lab quiz 2 terms. So this will be really helpful, I think, if you guys read through this. And I did it in order of the labs too. I didn't label like which term went with which lab. So just as you go through your labs, you can just look at this too. This kind of crystallizes the information a little bit better. Okay. Any questions on lab 23? Okay. Lab 24 was chemical methods of control, disinfectants, and antiseptics. So this is the one where we tested the lab's disinfectant compared with the wetting solution, I think. And do you remember so you have like a little tube of the disinfectant and then you put the bacteria in it and then you just waited. Do you remember, so do you know why we wait 2.5 minutes and then 5 minutes, 10 minutes, and then 20 minutes? Does that make sense?
The longer it sits in there, the more they kill.
Yeah, exactly. Or theoretically, anyway. That's what we're testing for. So, and And what is the difference between a disinfectant and an antiseptic? I think you guys might have talked about this in class too.
Mm-hmm.
There's a lot to remember.
I always think of ants on living skin, like they're crawling on your skin.
Oh.
So antiseptic is
Yeah.
living tissue.
Mm-hmm.
And then, what was the other?
Disinfectant.
Disinfectant, I'm dissing on that object, it's so dumb. So I just,
Whatever works. Yep. Yeah, and they're both considered antimicrobial, and they can be either bactericidal or bacteriostatic. Do you guys know? Do you remember the difference between bactericidal and bacteriostatic?
Cidal, it like kills it all, and then static, it just like slows the growth. or
Yeah,
stops
just
it.
kind of slows it down. Exactly. Do you guys have any questions on that lab? Well, do you remember which was more effective between the lab disinfectant and the wetting solution?
I couldn't come back with the results.
Oh, you can. Well, the wetting solution. Well, does anybody else remember? The wetting solution, I think, is what they used in the anatomy lab, right?
oh.
I don't know, but that was more of a bacteriostatic, I think. I don't know. There was still a lot of growth with the wetting solution. So the lab disinfectant was like 10% bleach, so it killed really well.
I think you're right, because it still degrades, but at a slower rate in the anatomy lab, I think.
Yeah.
Because isn't it like formaldehyde? I think
So. I don't know what it's made out of, but it's not as effective as bleach.
Love bleach.
It works.
Yay.
So lab 25 was chemical methods of control antimicrobial drugs. And this is definitely one that you're going to want to read through the background of. Because it talks about, let's see, is this the one that talks about Ignaz? No, this one talks about Pasteur and Alexander Fleming. And do you remember what Fleming did?
Discovered penicillin?
Yeah, and that was, yeah, so that's considered an antimicrobial drug now. And what is the clinical significance of the disc diffusion method? Like, how would you use it in a clinical way?
To see what will kill, or what will, oh shoot, which antibiotics will work on the microbe you're trying to kill. And then I guess from there you can also see how much to use.
Yeah, exactly. Like someone could possibly come in, sick, and you could isolate whatever bacteria is making them sick, put it on a plate, and then test out different antibiotics to see which one's going to work.
Because some might be resistant to some
Yeah.
antibiotics.
Yeah. There seems to be a lot of them out there nowadays. Yep.
Can I ask a question?
Oh, sure.
What bacteria is most resistant that is found in hospitals?
Pseudomonas?
Yep.
Good job. Okay.
Yeah, and it was interesting because in lab, a couple of people had Pseudomonas, and there was no zone of inhibition at all. It was just like growing everywhere. It didn't care that there was antibiotics. And do you remember the name of the special agar that we used in this lab? It is called Mueller-Hinton agar. I don't think you'll need to spell it, but you might need to identify it. You know what I mean? This is just special agar that allows the antimicrobial agents to diffuse freely. So that's why we use that. Definitely, yeah, read through that and make sure you know all the terms, what zone of inhibition means, minimum inhibitory concentration.
Kirby-Bauer test.
Kirby-Bauer test, as far as I can tell, it's the same thing as the disc diffusion method.
I
Do you know?
remember he talked about it. It's actually going to be on this next test. So
Oh, it
I mean,
is?
That would be very wise. The Kirby Bauer test, if I remember right, it just showed if the antibiotic was working, but the Mueller-Hilton with the Mueller-Hilton-Agar, but
Oh, okay.
isn't it? No, maybe that's the one where I'm trying to remember. Crap.
I think you're right. And then the E-test shows, isn't it the one where you can see
Like how
much to use? So you don't
Yeah.
overdo it.
Overprescribed.
I think you're right.
That's what the
Good
Kirby
job.
Bauer test is for?
Something, I don't know.
Isn't it? Crap,
I-
that's gonna bug me. Now I'm
Yeah,
gonna
when I
find
was
out.
reading through it, I really couldn't tell if there was a difference. To standardized Kirby Bauer test for Agar diffusion
The
methods.
E-test, is that what you said?
Yeah, I think that was the other one. And like in the picture on the slide had that, it's like a scale, and you set that,
Oh,
instead of the
yeah.
disc alone, you put the stick, it's like a stick with measurements on it.
Okay,
And
yeah.
depending how big the zone was, it would be,
Oh, you're right. Kirby Bauer Agar diffusion method.
I know
It's
enough
right
to be
here in the notes.
dangerous.
Oh, cool. What does it say in the notes?
Okay. If the chemotherapeutic agent is effective, a zone of inhibition is observed around the disc. A more advanced diffusion method is the E-test. Good job. Enables lab technicians to estimate the minimal inhibitory concentration, or the MIC. So the lowest antibiotic concentration that prevents visible bacterial growth.
Okay. This doesn't say anything about the E-test, so as far as lab
Oh,
goes.
yeah, that's true, that was from class. I
Yeah, you don't really need to know that
Never
for
mind.
just for the lab
Sorry.
quiz, but I mean,
Sorry,
in
brought
general,
that
you
up.
should know. It's
I'll
probably a
shut
good
up.
idea to know it. And this one was pretty straightforward. You might want to know that we swabbed the agar plate 3 times. Do you remember that? Just to make sure it really had a lot of bacteria on it. Would be good to know. And yeah, as far as that goes, I think that's all. Do you guys have any questions on that one? Okay, this one was the interesting one called, Lab 26 was the effectiveness of hand scrubbing.
This was gross.
I know. This was really interesting, I thought. I found it really interesting. Okay, so what's the difference between normal microbiota and transient microbiota?
Normal stays longer and then
Mhm.
transient like leaves after a little bit.
Right, transient can be there for days or weeks, but eventually it leaves, while normal microbiota is essentially there, always. Okay, and do you remember who Ignaz Semmelweis was and what his important discovery was?
I don't remember who he was, but he figured out to wash your hands between working on an autopsy and going to deliver a baby.
Yeah, yep.
That's not a good idea. You should wash your hands.
Yeah, exactly. Yep, he figured that out.
Like a Swedish doctor or something?
I think Vienna Hospital.
Ah.
Yeah, so he told people to wash their hands, and then that resulted in less puerperal sepsis. So I guess that's what happens during child delivery. And the death rate dropped from 12% to 1.2% in 1 year. So, very good discovery. So why do doctors scrub their hands so long before performing surgery?
Are they trying to get rid of that transient?
Yeah,
Microbe.
exactly. And do you know how long it takes to get rid of the transient
10
microbes? Yeah, 10 to 15 minutes. It says you have to just sit there and scrub with soap for that long. Seems like that would kill your hands, but do what you have to do. Okay, and then I think it would also be good to know, do you guys remember which soap was the most effective?